Hello, everybody, and welcome to today's webinar on sterility, manufacturing, and regulatory challenges in a constantly changing world of biopharmaceuticals. I'm Tim Wright, Editor of Contract Pharma, and today's session is being sponsored by both Nelson Labs and Sterigenics. Nelson Labs is a global provider of microbiological and analytical lab testing services for medtech and pharma companies. You can learn more about them at nelsonlabs.com. And Sterigenics is a global leader in comprehensive sterilization solutions, meeting industrial sterilization needs in the med device, pharma, advanced applications, commercial, and food industries. And you can also learn more about them at sterigenics.com. And we have three speakers on the line with us today. Jasbir Arora is Director of Bioanalytical, Regulatory, and Quality at the Jefferson Institute for Bioprocessing, along with setting up an analytical lab for characterization, quality control testing, and release assays for biopharmaceuticals, including cell and gene therapy. Jasbir teaches and trains both students and scientists from the biopharma industry. Bart Kroonenbergs is Technical Director of Irradiation at Sterigenics. He joined Sterigenics in 2005, and during his career with the company has performed IQ, OQ validation of gamma and electron beam irradiators, and designed and executed process definition programs as well as PQ dose map studies for customers, specifically those in the biologics and pharma industries. And lastly, we've got Karen Peters. She's the Senior E&L Expert at Nelson Labs. She joined Nelson Labs Europe in 2010 as a study director for different applications, and she now leads a team of study directors with a focus on large volume parenterals, ophthalmics, and disposables. She's presented on the setup and challenges of extractables and leachable studies for these applications at several international conferences. In today's session, our speakers are going to focus on product development, including sterilization and lab testing strategies to help ensure there's no contamination in the development and manufacturing of single-use systems to promote end-user safety. We will be accepting questions throughout the talk. You can send those in using the box at the bottom of your screen. Questions will be answered after the presentation in a brief Q&A. And this session will be archived on our website, contractpharma.com, for 1 year. If you're having any technical difficulties, you can type those into the question box as well, and someone on the back end here will help you out. At this point, I will turn it over to Jasbir.
Good morning, everyone. Good evening, good afternoon, depending upon where are you in any part of the world. So my name is Jasbir, and I will be talking about the initial part of the webinar. So you will be getting an overall feel about what is happening in the biopharmaceutical world right now, the changing scenarios of technological innovation, as well as the regulatory guidelines, especially based upon the quality by design approaches. And also we will be talking about giving you a very broad overview with my team members about the gamma radiation and sterilization, especially on single-use bioprocessing components. And we will be focusing on extractable and leachables on single-use components. We will be talking about the risk-based strategy, especially based upon ICHQ-9, focused on extractables and leachable. And at the end, we will be also talking about the process qualification, focusing on the gamma irradiation and sterilization. So overall, we will be talking about the sterilization, extractable and leachable testing, focus on the single-use bioprocessing components to promote end-user safety. So I will be giving you a brief background about what is happening in the biopharmaceutical world right now. But before we really talk about that, I will show you this slide, like we're talking about some regulatory guidelines, and I will give you a brief background of Jefferson Institute for Bioprocessing. Jefferson Institute for Bioprocessing, or we call it as JIB, is located around 30 miles from Philadelphia International Airport. We are in suburb of Philadelphia, and we are less than 3 years old. We were started, and then COVID happened, of course. But what we do at Jefferson Institute of Bioprocessing is that we work with different pharmaceutical companies, and we do different kind of process development studies, technology transfer, any kind of product evaluation services. That's what we do. We have a 25,000 square foot facility, which is going to be double in the next coming 2 years. And we have been also taking an initiative to expand our services to the GMP area in coming few years. Started solely on monoclonal antibodies, but now we have moved towards cell and gene therapy and next generation vaccines. And this slide gives you an overall summary of what we have been doing at Jefferson Institute for Bioprocessing, our process capabilities, operations, different therapeutic modalities. We have an analytical quality control lab as well. And we do also consulting. Being a part of Thomas Jefferson University, we also have master's program where we have students, they do their different kind of programs, but we have a master's program focused solely on biopharmaceutical processing and biopharmaceuticals. We have certifications, PhD programs, and also we work with different industry folks and do industry training because there is a huge demand right now in the area of biopharmaceuticals. Talking about different therapeutic modalities, which I have also listed on this slide, whether we have protein, monoclonal antibodies, viral vectors, cell and gene therapy, oligonucleotides, whatever we are manufacturing. So the overall process, the strategies, as far as the regulatory agencies are concerned, they don't change too much. The regulatory requirement, if you focus on CMC, they remain similar. No, they are not the same, but the approaches, but the strategies for CMC, they remain same. For example, if we are moving from phase two, from initial R&D or discovery or preclinical studies to the phase one, we talk about proteins. We don't have to characterize our protein fully. Of course, we need to know what we are working with. We have to have our analytical methods qualified. But really, when we are in phase three, when we are going to file our BLA, we need to have our methods validated. So in a similar way, if we talk about cell and gene therapy, that the concepts are similar when we are going for our manufacturing, when our process is being developed, when our product is being developed, we are getting that understanding. But one key factor when we are moving from our preclinical to phase one, doesn't matter which therapeutic modality we are working with, is the safety. Safety of the subjects, of the human subjects, is a key for FDA when we are moving from preclinical to phase one. And when we talk about safety, how we have to talk about the testing, the qualification, and the clearance of all the impurities and contaminants. That's a key which starts right in the beginning. Your analytical method may be, validation may be required at the end or in phase three, but the safety of the patient starts right in the beginning when we have first in human studies are being done, when our drug is going to touch humans for the first time. That's where all these factors come in. The contaminants, impurities have to be taken care of. So and that is also visible when we are in a commercial state. If we look at what is happening in the world right now, if we look at different kind of the citations which have been given by FDA, they have been in this area, they have been consistent. If we look at a few deficiencies from 2013 to 18, we see that 21 CFR 211-113B, which says that there should be appropriate written procedures which are designed to prevent the microbial contamination of drug products. And they have to be sterile. So that has to be established. And we see that they remain consistent from 13 to 2018. And also, if we look at some of the recent issues, what we have, all these 21 CFR 211.113B deficiency letters, they are not changing. Whether we are in 2020 or 2021, we are getting those citations as manufacturers. So it means that there is still room for improvement. There is still room for technological innovation, and as well as the regulatory guidelines. For example, USP 665, which talks about risk-based assessments. Now we have to come up with strategies which are risk-based. So we need to take care of the regulatory guidelines. We need to take care of our technological innovation and make sure we are moving in the right path. One of the innovation which we as a manufacturer have done is the use of single-use systems. Now, single-use systems have a lot of advantages when it comes to making our products sterile, keep our products sterile, which are going to go to humans. Now, what are single-use systems? Basically, we can talk about single-use system at two different levels. First level, what you are seeing right now, some examples. We have some disposable chromatography columns, some filter capsules, and a few other things which you see in the pictures right now. These are the assemblies. These are the components which are being used for the manufacturing of our drug substance and drug product, and then they are discarded at the end. We just use it for one run, and then they're discarded. So they have been really, really more popular for bioproduction, but they have also been used for small molecule drug as well. So they have been used. So that's a huge, there are a lot of advantages of using our single-use system. Now, those are the things which we have seen in the previous slide, like the smaller components. We can have our single-use system throughout the biopharmaceutical manufacturing, and this is the slide which you're seeing right now from end-to-end manufacturing of a protein monoclonal antibody. In fact, this is what the system what we have at Jefferson Institute of Bioprocessing, which is whole single-use Cytiva Flex Factory. So we have, in the beginning, whether we are doing cell expansion, when we are doing cell production, we can use bags, sensors for our whole manufacturing operations. When it comes to chromatography, we can use different kind of resins, column. So we are using them for one run. So after that, we can discard that. So these are the different components of single-use system which are being used throughout the manufacturing process. But like any other new innovations, there are a lot of advantages of using single-use system, as I said. You talk about clean in place, team in place, you get rid of all those issues when you're using a single-use system. So there is elimination of cleaning and sterilization processes. The ease there's a lot of ease when we're using this system. These are cost effective, and our validation processes become really easy. And what we have seen using single-use system, that it reduces the risk of contamination. There's increased flexibility. There's a time saving. So we are competing against time. We want to get our drugs into the market as soon as possible. So there is a huge time saving between different production batches. Even if you want to switch from one particular product to another, the switching is relatively easier as compared to when we are not using single-use system. But like any other new technologies, we will also have challenges and issues. We have the same issues with single-use system as well. So there can be issues where there are extractable and leachables from the components which we are using. And in fact, this is a huge issue of extractable and leachables. And also, what about different vendors? If we switch from one vendor to another, there can be issues of extractable and leachable, because we may have done our validation with one vendor, but if the things change from one vendor to another, then the uniformity is lost. So we are thinking about more work at that stage. Then there can be an issue with the relationship with different vendors. So what if there is no consistency from one vendor? So we will have to make sure that the vendor with whom we are working has been thoroughly qualified. And if we have to make a change, there is a proper change management system. So there are issues with single-use system as well, which can be taken care of through different strategies. Now, FDA has come up with a 2011 guideline with the concept called quality by design. There's a process validation guideline which came in 2011. The first guideline came in 1987, and then the second came in 2011, which talks about the concept of quality by design. Now, what is QBD? QBD is basically, we are saying that we just cannot ensure the safety of our patients by doing end product testing. Only by doing some release assays is not ensuring that our product is safe. We have to build quality in our processes, in our product. We have to make sure when we are manufacturing, when we have different unit operations, the quality is built into it. Now, there are a lot of different components. There are different regulatory guidelines which comes into quality by design concept. And the ICH-Q8 is a starting point for that. It starts about the pharmaceutical development. ICH-Q9, the key component of quality by design is the risk assessment strategies. Again, we have the ICH-Q10 pharmaceutical quality system and ICH-Q11 development and manufacturing of the drug substance. What we are going to do today is that we talk about the two important components based upon ICH-Q8 and ICH-Q9. ICH-Q9, when we talk about sterilization issues, how we can go with a different risk assessment strategies based upon quality risk management, ICH-Q9. When we have to build quality into your product, how the gamma irradiation can come there and help us to make sure that we are building quality into product. We are making sure that our product is not contaminated. So these are the two important topics which we're going to cover in the next 30 minutes or so. And my colleagues, Dr. Bart Tornbergs and also Karen Peters, they're going to talk about these two issues. First, we are going to talk about the gamma irradiation, especially focused upon cobalt-60. And then we will talk about different risk assessment strategies. And at the end, Bart will again talk about the process qualifications. So I will now hand it over the stage to Bart so he can talk about the initial introduction of cobalt-60.
Thank you very much, Jasbir, for the introduction and for setting the scene that is describing the world of biopharmaceuticals, and then particularly the single-use bioprocessing components. As you righteously pointed out, sterilization is a key aspect that should be taking place prior to using these. And I'm going to spend the next 5 minutes or so just using 5 slides to present a high-level overview of probably the dominant outsourced sterilization modality that is used for these bioprocessing components, which is gamma sterilization using cobalt-60. First of all, let's look on a really high level, very generically, gamma radiation, without going into specific isotopes in this case. Looking here on the left-hand side, gamma radiation is a form of electromagnetic radiation. You can see the electromagnetic spectrum presented there, taken from a textbook, high school textbook. And you can see if you go from the left-hand side in that image to the right-hand side, that the wavelength of the wave is becoming shorter and shorter. So its energy is increasing as you go further to the right-hand side. You're crossing the barrier between non-ionizing and ionizing radiation at the end of the visual spectrum. So everything on the right-hand side is ionizing, and you can see that gamma radiation spans a certain range of wavelengths, but it's comprising radiation, electromagnetic radiation that is coming out of nuclei, out of radioactive species. You go to the right-hand side, passing through matter of this type of radiation is generally quite well, and it is always characterized by an exponential decrease of intensity with thickness. So depending on the isotope used, you can have better or worse penetrating characteristics, but in general, it's always penetrating better than its sister radiation type electrons, which is identified here. You can clearly see the difference in the image where you need lead pieces to shield the radiation flux for photons, whereas a little piece of aluminum can already do that in most cases when the radiation is comprising electrons. More specifically, changing towards cobalt-60, which is the nucleus of choice already since the start of the radiation processing industry. Left-hand side, you can see the penetration characteristics of the radiation field when it's hitting something of homogeneous nature. On the y-axis is the percentage of intensity of the radiation flux, which is then expressed versus the depth of penetration in centimeters of water. So if you're looking, for instance, the penetration characteristic of a Europallet, 80 centimeter of depth of 0.1 gram per cc bulk density, that is the equivalent of 8 centimeters of water, and you can see that the intensity of the radiation flux is reduced to about 2/3 of what it had at the surface, which is still manageable in reality. So if you look at this point, penetration through material, and then presenting the product to the radiation field in different manners, as for instance visualized here in general by the turkey, which is presented to radiation field as well, and is spinning in front of the radiation source in this case, so that its surface is cooked, but not black, and its inner piece is not red anymore. It's very similar in nature what we do in a gamma radiator, using the isotropic nature of the radiation field. Isotropic means there's no directional preference. The radiation is emitted in all 3 directions, as is shown schematically here in this image on the right-hand side, shown with the ionizing source of radiation in the very center, the blue rectangle. And then you can see the intensity of the radiation field is getting less and less as you progress further. Standoff distance is increased with respect to the source. You first have a bold line, the line becomes thinner and thinner, it becomes dashed, and then in the end it becomes dotted. So in general, The technology lends itself inherently very well to take into account product variability, as could be the case for bioprocessing components if you just look at them from density perspective, so density in an irradiation container. The process variables are just the pathway you take through the irradiator, how much time you spend in the irradiator, how you load the product in the irradiation container, and the mixing in the irradiator. Typically, processing takes a couple of hours. It's non-adiabatic heating, where the peak temperature depends on the irradiator, but can be 40, 45, maybe sometimes 50 degrees Celsius. And typically, there's a lot of product irradiated concurrently. How does that look real life? A schematic cut through of an existing gamma radiator in our network is shown here. It's a modern irradiator. I'm not going to say too much about this, just that if you look on the inner component inside of the concrete, which is the part where the blue arrow starts, that cut is presented in a horizontal way there. And you can see there are many rectangles, where each rectangle presents a position that product takes in the irradiator. So there are in this case a total of 36 positions in the irradiator, where each position has an irradiation container that is the size of half an industrial pallet. So at any point in time, there are 18 industrial pallets worth of product in the irradiator, irradiated concurrently. So it's a quite large machine that we're talking about here. Material compatibility for radiation sterilization. If you look at the AMI TIR 17, it's the Association for the Advancement of Medical Instrumentation. It's a non-profit organization that is involved in developing standards and guidance documents. They issued in 2017 like a compatibility overview of commonly used materials subject to sterilization modalities. This picture is taken from their booklet, and you can see here listed from top to bottom are a list of commonly used medical plastics, can also be used for bioprocessing components if you want. And on the x-axis, you can see the scale with increasing absorbed dose. And then you can see there are actually the equivalents of green, orange, red, traffic light analogy, where compatibility is expressed as a white, transparent part, then orange, used with caution, you have the hatches from top right to bottom left, and not recommended for going into that range of absorbed dose is expressed here as a hash from top left to bottom right. You can see that for a lot of the typically used plastics, across the typical range of sterilization processing, which goes up to say 40 or 50 kilogray, many of these plastics are either transparent in this scale or in the use with caution. This means that radiation sterilization is possible for a substantial range of polymers, also the ones that are very commonly used. And if you combine that with the penetration capability explained before and the availability of gamma radiators across the globe, gamma sterilization is, I can say, the dominant outsourced modality for single-use bioprocessing systems. Looking at the validation of that sterilization process, this is an schematic extract taken from ISO 1137-1, which is the radiation sterilization standard for medical devices. You can see as an input, left-hand side, sterility assurance level, how clean does the product have to be after the sterilization step. And then you have the product definition, which describes how the product is manufactured and including its packaging system. Then the validation steps are presented into the dotted part here, which comprise, first of all, process definition. So you establish the minimum dose needed to render the product sterile, which can be based on bioburden and its resistance to radiation. Also, you have to establish the upper limit for any kind of sterilization process. So the maximum acceptable dose. So up to that point, all of the critical performance attributes of the products are still maintained. Next, you have process validation, which is establishing an irradiation process in the selected irradiator, so you can be assured that you will always render product irradiated within the established dose specification. In all of these steps, the critical challenge will be how to handle product variability. And it's about this that the next two contributions in this webinar will focus. First one will be by Karen in process definition step, extractables, leachables, and after that I will be turning back to you. I will be talking a bit about process validation.
Okay, thank you, Bart. In this part of the webinar, we will be talking about extractables and leachables studies on single-use systems used in bioproduction. So there are several guidelines that mention that manufacturing equipment or its impact should be evaluated. For instance, the Title 21 of the Code of Federal Regulations states that surfaces that contact components in process materials or drug products shall not be reactive, additive, or absorptive so as to alter safety, identity, strength, quality, or purity of the drug product. And similar statements are also made in ICH 7 and in Good Manufacturing Practices of the EU. So these guidelines mention that manufacturing equipment should be assessed, but they don't mention how this assessment should be done or what type of testing should be performed. Currently, the United States Pharmacopoeia is drafting two chapters, USP 665 and USP 1665. The USP 665 will contain a standardized extractables protocol for plastic materials, components, and systems used in the manufacturing of pharmaceutical drug products and biopharmaceutical drug substance and products. In the United States Pharmacopoeia Chapter 1665, this chapter will contain more background information and also an example of a risk evaluation matrix. Both these chapters have been published in draft already a few times in the Pharmacopoeial Forum, and they are now expected to become officially in May of next year, so 2022. Since the guidelines in the past were quite vague on how to test for extractables and leachables for single-use systems, there have been multiple interest groups that have been working on this topic. And one of the interest groups that first made some publications or recommendations for that is the BPSA, or the Bioprocess System Alliance. So this was originally mainly a trade association of suppliers of single-use systems and later on also end users of single-use bioprocess technologies joined. They made two public recommendations for extractables and leachables testing. Another group, interest group that has become quite famous on the topic of extractables and leachables is the BPOG, or the Biopharm Operations Group. This was originally an association of end users of single-use systems, and then later on also suppliers joined. The first publication they made was a standardized extractables testing protocol in 2014. So this was a quite elaborate testing protocol. It contained 6 different extraction solvents and 4 different time points to be tested. And that caused quite some resistance with the suppliers of single-use systems because they thought the standardized protocol was too elaborate to apply on all of their components. In 2017, this group published a best practice guide for evaluating leachables risk. And then in April of last year, 2020, they published a revised best practice guide for extractables testing, where they downscaled the testing protocol. And this was backed up with test data that they published in September of that year. So if customers come to Nelson Labs and they want to test their single-use systems for extractables and leachables, we advise to include the following 4 steps. So first, we advise to perform a risk assessment of the materials used in the production process. And then in a second step, extractables data can be gathered for the materials that are of high risk. Then the extractables data can be evaluated, and it can be evaluated whether a leachable study, a subsequent leachable study is necessary. So I start with a risk assessment. The bioproduction process may contain a lot of different single-use systems, which may be custom-made and may contain components of different suppliers. And if an extractable study would need to be performed for each of those components, this would be a huge task. So instead of testing every single-use system or single-use system component for extractables, a risk-based approach can be applied to focus on those materials which have the highest impact. So the goal of the risk assessment is to select those components which have the highest impact on the safety and the quality of the final drug product or on the process performance. So if you want to perform a risk assessment, first you need to create a list of all the product contact materials. And it's very important to understand your manufacturing process very well from start to finish to make sure that all materials are included. This may also include people from different departments to contribute. Then once this list of product contact materials is available, these materials need to be assessed versus a number of risk factors. So this slide shows an example of some risk factors that can be taken into account, such as material compatibility, proximity to the final drug product, composition of the contact solution, the surface area to volume ratio, the contact temperature and contact time, and pre-treatment steps such as sterilization. So the BIPOC guide for leachables testing includes an example of such a risk assessment. And this protocol includes 5 different risk factors. For each risk factor, 4 different risk levels with a rating are defined and a weight factor is defined. And by making a combination of the different risk level ratings with a weight factor, an overall risk rating can be calculated. And this overall risk rating will then help in classifying whether a material is high risk, moderate risk, or low risk. Also, the USP 1665 includes an example of a risk evaluation matrix, and there the risk evaluation is a 3-step process. In the first step, a value is defined for each risk dimension. So there are 4 different risk dimensions specified and 3 different levels. And this will result in a numerical risk sequence for the component. For instance, if we take an example of a sterilization filter that is in short time contact with the process stream, this results then in a level 1 for duration of contact. At ambient temperature, this results in level 2 for temperature of contact. If the contact solution is highly organic, this will result in a level 3 for chemical composition of the process stream. And if we assume that the chemical composition of the filter is of high risk, we also have a level 3 in the last dimension for chemical composition of the component. So the numerical sequence then becomes 1, 2, 3, 3. But the sequence needs to be given in order of decreasing digit values. So the numerical risk sequence will be 3, 3, 2, 1. In step 2, that numerical risk sequence is linked to a level of characterization, and that link is made in a table that's included in USP 1665. And for our example, for a numerical risk sequence of 3, 3, 2, 1, you end up with a level B or level C, that is moderate risk or high risk, and it depends on the risk dimension that scores to what the risk level is. And in our example, it was temperature that had a level 2 score, and so our component is scored as a level C or a high-risk material. In the last step, mitigation factors may be used to adjust the characterization level downwards. A first mitigation factor that can be used is clearance after the contact processing step. So is there a next processing step that might eliminate or reduce the amount of leachables in the contact solution? In our example, we are looking at a sterilization filter, which is used in the final sterilization filling line. And so there is no next step that might remove or reduce leachables. So this mitigation factor is not applicable in our example. The second mitigation factor is the clinical use of the final drug product. If the duration of the treatment of the drug product is less than 7 days, if the daily dose is less than 10 milliliters, or if the drug product is dosed orally, then the level C testing can be reduced to a level B. So in this slide, a table is shown from USP 1665, where the risk level is linked to the level of testing that needs to be performed. So for level A or low risk materials, a 50% ethanol extract needs to be prepared and tested for non-volatile residue and UV absorbance. For level B or moderate risk materials, 50% ethanol extract needs to be screened for organic compounds. If our material is a level C or high risk material, 3 different extraction solvents need to be used: a 50% ethanol extract, a water extract at low pH, and or a phosphate buffer at pH 10. And all 3 of these solutions need to be screened for organic extractables and or for elements as deemed necessary. So then we arrive at the real extractables and leachables testing program, because for those materials that were risked as a high risk, extractables testing would need to be performed. In some cases, the supplier might have extractables data available. If the supplier doesn't have extractable data available, it is the responsibility of the end user to demonstrate that the single-use system is suitable for the end application and that it does not alter the quality or safety of its product. Because the end users sometimes want to compare extractables data of different suppliers, there is an increasing demand for standardized extractables protocols for extractables testing performed by the supplier. And such a protocol would need to cover the majority of biopharmaceutical applications, and it would make it more easy for the end users to compare data from different suppliers. Because the setup of an extractable study has a very high impact on the outcome of the extractable study. So when one wants to compare extractables data from different suppliers, it's very important to evaluate whether the same setup or a similar setup has been used. So as mentioned earlier, the BIPOC has published such a standardized extractables protocol. On this slide, a table of that protocol is shown as it was published in 2020, so the adjusted protocol, where they already aligned more with USP 665. So this latest protocol includes 4 different extraction solvents, and depending on the component, 2 or 3 different time points need to be included. So also USP 665 contains a standardized extraction protocol. The extraction solutions were already mentioned earlier in the slides. And per component, only 1 time point is specified for testing. So components that are only in short contact with the process stream, extraction of 24 hours at 40 degrees is proposed. For aseptic connectors or disconnectors, 7 days is is proposed, and for components that are used for longer time storage, 21 days or longer is proposed. So once the extractables data is available, the data needs to be evaluated. If extractables data is obtained from the supplier or another source, it's always important to evaluate whether the data is suitable for the intended application. One of the aspects that needs to be verified is the extraction solvents. Do they cover the organic content, the pH, and the polarity of the actual contact solution? Were conditions of time and temperature covered in the extractable study? And was the material subjected to pretreatment steps such as sterilization? Because especially gamma irradiation, for instance, is a high energetic sterilization technique, and this might alter the extractables profile of a component or of a material. It's also important to look at the analytical techniques. Were screening analysis used, and did they use a combination of different analytical techniques to screen for a broad, wide range of different compounds? An analytical evaluation threshold can be calculated to check the sensitivity of the methods, and this analytical evaluation threshold can also be used to evaluate the extractables results. If the results of the extractable study and the concentrations are all below the analytical evaluation threshold, the component can generally be considered to be safe. However, there are some exceptions if for final storage containers for the final drug product, and also for components that are used in the final filling line because they make direct contact with the final drug product, it is recommended to always perform a leachable study. And then now I will hand over the word again to Bart. We will talk more about process validation.
Thank you, Karen, for sharing these practices around ENL as they apply to bioprocessing single-use components. That is part of process definition. We go back to this slide here. So process definition is establishing the specification of absorbed dose so that you know the minimum required dose that you need to exceed in order to make a sterile label claim, and at the same time maximum acceptable dose, the level that you cannot go above, or one or more of the critical performance attributes, including possibly ENL, will no longer be meeting the requirements. Now we go one step further, so assuming that you have established the dose specification for the product and that you have selected an irradiator of choice, how are you then going to move about getting the processing validated for that irradiator? Again, looking at the challenge that the variability in the single-use bioprocessing components brings to this process. So we jump to ISO 1137-1 again, which is the medical device sterilization standard. We are then ending up in section 9.3, performance qualification, PQ dose mapping. So what are you then going to do? You're going to define or determine the dose delivery characteristics for defined product in a defined configuration in an irradiation container that you move then about in a very specific pathway through the irradiator. What are you obtaining? You're obtaining the zones at which the minimum and the maximum doses are concentrated. You're going to characterize the variation of the process, including the variability in dose extremes, so minimum and maximum dose. You're going to define a process monitoring practice for routine processing. And in reality, what does this mean? This means that you're going to put a lot of dosimeters in product in that defined load configuration, and you move it through the irradiator in that specified pathway, and then you're going to look at these dose measurements and you're going to analyze them. So if you're then going to look at product variability, so first upper axis homogeneous, non-homogeneous means within a carton, and then vertically between cartons, so variable or consistent. So easiest is if a product looks homogeneous within a box, and every box looks the same. That's very easy to then qualify in a gamma irradiator. An example could be pharmaceutical packaging, and typically within Sterigenics, these customers are onboarded on a daily basis autonomously by our facilities that are operating their irradiators, so without really corporate involvement. If you're taking one more box and now the product within a carton is non-homogeneous, but it's still looking the same box to box, for instance, could be a drug product, then this will involve corporate, as this is more complicated to onboard, more complicated to measure and to predict the absorbed doses. If you're going to look homogeneous within a box, but variable box to box, that could be single-use bioprocessing components, non-homogeneous within a box, and at the same time different box to box, same challenge can come from bioprocessing single-use components. So all three, you need to take special care for performance qualification dose mapping. So now I have two examples for bioprocessing components. First example, homogeneous density within a box, but box to box, there's a lot of variability in the mass, comprising a bulk density range nearly a factor of 3 between the lowest density and the greatest density. And the desire of the customer is to mix all of these different boxes together to the extent possible within the same irradiation container. Radiation container and load configuration shown on the left-hand side is the irradiation container coming from the irradiator that I talked about 10 or 15 minutes ago. You can see the fillets really nice. But then the question is posed, what are you going to dose map? Are you going to dose map the extremes, like product of only 0.05 gram per cc density? You dose map that. And similarly, you do the greatest density challenge? You could. Are you going to do more, where you're going to spot pick some boxes and make them of intermediate density or mix the lowest and the greatest together? You can be as creative as you want. But generally, the same issue resides, which is not necessarily in the validation step, as yes, doing only the extremes will be appropriate. It's more the problem of ongoing routine monitoring. Because on a routine basis, we are not keeping track of every single box, where it is put, how much it weighs. So we don't really know what to expect from those measurements made at specific positions at specific irradiation containers. Unless we are, for instance, running in order of increasing weight, which also is a bit of a burden from operational perspective. A solution is actually if you do everything that we just talked about and add one more position of those measurements in your grid, which is here indicated by the orange star, which is the top corner of the irradiation container. Between that position and the product, there's enough space, empty space, void, which makes the dose that you measure on that position identified by the star does not depend on what you're actually loading in the irradiation container. So it's a process monitoring position where you monitor in essence that the irradiation container was filled to the right height, that the pathway through the irradiator was respected, and that the time in the irradiator was also respected. Concept is very similar to what you do in an electron beam irradiation process. It's a very elegant way, and then you can just dose map the extremes at one position, and upon successful completion, you can then mix anything in this irradiation container going forward. Example 2, adding one more complexity, which is the non-homogeneous density distribution within a box. So the concepts of example 1 do apply. So you're going to try to map out the extremes and add that single monitoring position that will be used for routine monitoring. And then you have the additional consideration that you can either have empty spaces within a box, or for instance, the presence of metal components in substantial mass as they could come, for instance, from impellers. What you then do is you typically modify the dosimeter grid. You can add specific monitoring positions at or near these high density, high mass components. And you also have to be cognizant of the fact that the temperature increase on metallic parts can really be substantially greater than on plastic parts, which could also affect the measurement system. So in general, modifications made to the PTO dosimeter grid, and also maybe you're going to map out multiple configurations. The key takeaways is like either product looks the same from box to box, and is considered to be homogeneous within the box from density distribution perspective, then you can really easily predict what's going to happen from dose distribution and dose magnitude perspective, and it's actually quite pretty straightforward to do the PQ dose mapping. If the product is non-homogeneous within the box and or every box looks different, typically the PQ dose mapping process becomes more complicated, and you end up adding dosimeters for measuring the product dose and possibly to establish your practice for routine monitoring. Possibly also multiple configurations are then needed to be included, depending again on the complexity of the variability. On the right-hand side here, these are the typical challenges imposed by the majority of the single-use bioprocessing components. But we have global harmonized practices in place for tackling these challenges. Overall, the key takeaways from this webinar going in were to try to give you a basic understanding of the current developments in the use, sterilization, and ENL testing of single-use bioprocessing components. And throughout the webinar, I believe we have achieved that. So first, we had Jasbir giving a presentation on the world of biopharmaceuticals, and in particular, the focus on the use of the single-use bioprocessing components. We had an introduction about gamma radiators, and then more in depth about ENL, how to apply an ENL management program on these components. And then finally, what goes on in the typical gamma radiator after process definition took place. So how to move to a successful PQ dose mapping exercise. That said, I would like to turn the word again to Tim.
Yeah, great. Thanks so much, Bart. Really terrific job. You, Karen, Jasbir, well done, and covered a lot of ground there. So I'm going to go ahead and conclude today's session. Thank you, Karen, Bart, and Jasbir for a really informative talk. Thank you to today's session sponsor, Nelson and Sterigenics. Thank you so much, everybody in the audience, for your attendance and participation. And this session will be archived on our website, contractpharma.com, for 1 year after today's date, if you'd like to check it out and share with your colleagues. And thank you very much for your attendance. Appreciate it. Have a great rest of your day, everybody. Take care.